Assalamu alaikum everyone. In this video lecture, we will be discussing paper 1 or multiple choice question paper of O-level biology 5090. So this is a sample paper. From sample paper I mean that this paper is created according to the new O-level biology syllabus. You all know that the O-level biology syllabus has been changed, and in your examination from June 23 you will be tested according to the new syllabus. So I have made several sample papers according to the new syllabus, and this is the second sample paper that we are discussing. Let's start. MCQ number 1. Lichens are formed from two different organisms living together. Organism X and organism Y are found in most lichens. So guys, they are saying that lichens are formed from two organisms which live together, and these are organism X and Y. The table shows some of the characteristics of organism X and organism Y. Which kingdoms do X and Y belong to? Okay, so we have to identify the kingdoms of the organisms X and Y. If we look at the description of X, it says that it's made up of strands called hyphae. So guys, hyphae are only found in the multicellular fungi, those fungi which are made up of many cells. Hyphae has cell walls and many nuclei. So guys, X is definitely a fungus. Mainly we can say a multicellular fungus. If we talk about Y, so Y is a single celled organism, and cell contains nucleus and chloroplasts. So guys, this is most likely an algae. So always remember this point, that if you find an organism which is single celled and if it contains chloroplasts and nucleus, it means that that organism is an algae, which is a microscopic plant, and this belongs to a kingdom which is known as prototist kingdom or kingdom prototista. So why is a prototist? The answer is B. Let's move on to MCQ number 2. Which statement about the structure of DNA is correct? Base A always pairs with base C. No guys, you should know that A pairs with T by the formation of two hydrogen bonds, and C pairs with G by the formation of three hydrogen bonds. So A pairs with T and C pairs with G. Alright, base A always pairs with base T. This is the correct answer. DNA is made up of protein. No, DNA is made up of nucleotides, right? DNA is made up of nucleotides and not proteins. You need to remember that. Proteins are made up of amino acids. DNA forms a single helix. No, DNA forms a double helix. You should know that DNA is double stranded, right? So other options are wrong and B is correct. Alright, you can remember the base pairing by using the mnemonics AT and GC for gold chain, right? MCQ number 3. 4 freshly cut potato cylinders were soaked for 1 hour in different salt solutions. So what we have done is that we have put 4 freshly cut potato cylinders into different salt solutions. The potato cylinders were then pinned to cork blocks. 2 of the potato cylinders are shown. So guys, what they have done is that they soaked 4 freshly cut potato cylinders into different salt solutions, and then after soaking them, they pinned the potato cylinders to a cork block, and 2 potato cylinders are shown. One is turgid and the other one is flaccid. Which solution would cause the potato cylinder to be most flaccid? So guys, what is meant by this statement? We need to pick up a solution which would cause the potato cylinder to become most flaccid. What is meant by most flaccid? This means that most water is lost. Most water is lost from the potato. Most water is lost from potato. What is actually meant by flaccid? Flaccid is a state of a tissue in which the cells they loosen because of the loss of water. So most flaccid means that the potato has lost most water. If the potato has lost most water, so it was placed in the most concentrated solution. You need to remember that if the potato has to become the most flaccid, if the potato has to lose most water, it should be placed in most concentrated solution, right? Because you know that higher the concentration, lower the water potential. So placed in most concentrated solution. So it should be placed in a solution with least water potential. We can say placed in most concentrated solution, or in a solution with least water potential. In a solution with least water potential, right? So if potato has to lose a lot of water, it should be placed in a solution which has the least water potential, so it loses a lot of water, and the solution which has least water potential is the most concentrated. So the answer will be D, that is 1.0 mole per dm cube salt solution, which is the most concentrated, and it will cause the potato to become most flaccid. Let's move on to MCQ number 4. Roots and leaves both act as sources and sinks for sucrose and amino acids at different times during the year. So guys, you need to know what is meant by source and what is meant by sink. Source is a site where sucrose is loaded. Source is a site where sucrose is loaded in phloem, and sink is the site where sucrose is unloaded from the phloem, where sucrose is unloaded from phloem. So as you all know that phloem vessels transport sucrose and amino acids, and transportation is always from source to sink, because at the source sucrose is loaded and at the sink sucrose is unloaded from the phloem, right? It's just like phloem vessels are just like trucks, just like the cartons are loaded into trucks at a source, for example factory. If we talk about the lace factory, and if the lace carton are being loaded into the truck, they will be loaded at the factory, which is the source actually. Then the truck will carry those chips cartons or lace cartons to the sink. Sink is the site where those chips will be unloaded, right? And sink is basically the site where those chips will be required, will be sold, right? So actually in plants, sources are the sites where sucrose is produced or is stored, and at source sucrose is loaded into the phloem sieve tube or in phloem vessels, right? In the sink, at the sink, sucrose is unloaded from the phloem vessels, right? So transportation of sucrose always occurs from sources to sinks, right? So they're saying that roots and leaves, they both can act as sources. Let me tell you how. That when the leaves are photosynthesizing, they are producing a lot of sugars and they are producing sucrose. So they become the source. And what these leaves can do, these can transport the sucrose to the roots, right? Sucrose can be transported from the leaves to the roots via the phloem vessels, right? So leaves in this case will be source and roots in this case will be the sink. When in some seasons when leaves fall and the new leaves are developing, so at that time the leaves are not photosynthesizing, and the sucrose stored in the roots is provided to the growing leaves. So in that case, roots will become the source and leaves will become the sink, right? So they're simply saying that roots and leaves, they both act as sources and sinks for sucrose and amino acid at different times during the year. At which point in the year are roots most active as source? So we have to select the option which provides us a point in the year during which the roots are most active as sources, which means that roots are providing sucrose. Sucrose is transported away from the roots, and that only happens when the new leaves start to grow, right? Because when the new leaves are growing in spring season, they are not photosynthesizing and they need carbohydrates and energy to grow, and the carbohydrates like sucrose come from the roots. So the answer is A. Let's move on to MCQ number 5. What happens to the heart valves when the ventricles contract? So guys, you should know that whenever the ventricles contract, what happens is that whenever the ventricles contract, for example, the right ventricle and the left ventricle, the blood can be pumped into the right atrium and left atrium, but this is the wrong direction. So for that, there's a valve which closes to prevent the backflow of blood, right? When the ventricles contract, one possibility of blood flow is to the atria, right? To the right and the left atrium. And this is, however, prevented by the closure of what? By the closure of which valves? The valves between the atria and the ventricles, and these are called the atrioventricular valves. So the backflow of the blood is prevented, right? By the closure of valves. Which valves close? The AV valves. So always remember this point that whenever the ventricles contract, the atrioventricular valves which are between the atria and the ventricles, they close. And these are bicuspid and tricuspid valves. Bicuspid valve is on the left side and tricuspid valve is on the right side. Another possibility of blood flow is that when the right ventricle contracts, the blood flows into the pulmonary arteries, and when the left ventricle contracts, the blood flows into the aorta. And for that, these are the right directions of blood flow, so the valves will open. And which valves actually open? Semilunar valves, right? So you need to remember this, that whenever ventricles are contracting, the atrioventricular valves, they close, and the semilunar valves, they open up. The answer is B. Let's move on to MCQ number 6. What happens to the muscles in the iris to make the pupil smaller? So guys, for this, you need to remember the rule of 3 C's. Rule of 3 C's. So always remember this point, that whenever the pupil has to become smaller, right, it means that pupil has to constrict, right? Constriction of pupil. What is meant by pupil becoming smaller? Pupil is basically an opening which allows the light to enter the eye, right? And if the pupil has to become smaller, or if the pupil has to constrict, what should happen? For the constriction of pupil, the circular muscles should contract. So always remember the rule of 3 C's. That is, constriction of pupil requires contraction of circular muscles. So contraction of circular muscle causes constriction of the pupil. So circular muscles will contract. And what about the radial muscles? Guys, you need to know that iris is basically the colored part of the eye. And iris contains 2 types of muscles, circular muscles and radial muscles. The iris is involved in controlling the pupil size or diameter. And whenever iris has to decrease the pupil diameter, it causes the contraction of circular muscles. And whenever circular muscles contract, the radial muscles relax. The answer is B. Circular muscles and radial muscles are antagonistic. If circular muscles are contracting, the radial muscles are relaxing. If the radial muscles are contracting, circular muscles are relaxing. So this is the most important point to remember, that both of these muscles are antagonistic. If we talk about pupillary dilation, right? If I talk about pupillary dilation, that is when the pupil gets larger. So in that case, what will happen? Circular muscles will relax and radial muscles contract, right? Opposite will occur. Let's move on to MCQ number 7. The diagram with the structure labeled X shows a bacterium with proteins on its surface. The diagram labeled Y shows proteins made by the human body. So guys, we can see that this is the bacterium, and it has protein molecules on its surface, and these protein molecules on the surface of the bacterium are called antigens. You all know that the molecules on the surface of the cell are known as antigens, and antigens are used to recognize a cell. The proteins are produced by the human body, and these proteins are called proteins Y. Which row shows the correct combination for destroying the bacterium? So guys, you all know that to destroy the bacterium, antibodies must bind to the antigens on the bacterium. So antibodies should bind to the antigens on the bacterium. This is the antigen, and to this antigen the antibody must bind. And when these antibodies bind to the antigens, what happens is that this bacterium is engulfed or phagocytosed by the phagocyte. So name of X is the antigen, right? And name of Y is the antibody, and the correct antibody or the correct shape of the antibody is this and not this one. Let me tell you why. Because the antibody should be complementary to the antigen. And what actually I mean by complementary, that the antigen must fit into the antibody, just like the substrate fits into the enzyme. So the shape should not be same, the shape should be complementary. And this is very, very important point. The answer is A. Let's move on to MCQ number 8. A student used two seedlings X and Y to investigate phototropism. The diagram shows their investigation. Okay, so in this investigation, students have used two seedlings X and Y, right? The diagram shows their investigation. So what the student has done is that in case of X, the student has placed a piece of glass at the shaded part of the shoot, right? Or the shaded part of the seedling. This is the shaded part of the seedling, and this is the part which is exposed to light, or we can say bright part. What happens is that the shoot does not bend towards light or does not grow towards light. And that's because, as you all know, what is the mechanism of positive phototropism? What is the mechanism? That the auxins, what auxins do, that they accumulate at the shaded side and stimulate growth at the shaded side. So the cells at the shaded side grow and cause the bending of the shoot towards light. This is phototropism, right? Positive phototropism. That the auxins accumulate in the shaded part of the shoot, and the shoot grows, or the cells of the shoot grows at the shaded part, and the shoot bends towards light. Over here, the shoot is not bending. You can see that the shoot stays straight and is not bending. And why is that so? Because there's a piece of glass which prevents the diffusion of auxins downwards. You need to know that auxins are produced at the shoot tip and they diffuse downwards. So what is preventing the diffusion of auxins? That is the piece of the glass. Over here we can see, in case of Y seedling, the piece of glass is inserted into the area which is exposed to light. That is known as the seedling facing the light, right? So over here you can see the bending. And why is that so? Because auxins will collect at the shaded part and the auxins can diffuse downward. And this can cause the growth of the cells in the shaded area and the bending of the seedling towards the light. So you can see the bending of seedling towards light. Which statement explains the difference in the results between X and Y? So guys, what should be the answer? The piece of glass in X stopped the auxin diffusing down the shaded side of the seedling. The piece of glass in X stopped the auxin diffusing down the shaded side of the seedling. This piece of glass is preventing the auxins to diffuse downwards, right? And that's why the seedling is not bending towards light. The answer is C. Let's move on to the next MCQ, MCQ number 9. The diagram shows part of the mechanism that controls blood sugar concentration. A person does 1 hour of exercise, starting with the pancreas. What is the sequence of events in which the hormone glucagon is involved? So, guys, the person did 1 hour of exercise, and you need to know that whenever you exercise, what happens? The blood glucose concentration falls, and this is detected by the islets of Langerhans and pancreas. And pancreas respond by secreting glucagon, and glucagon is released into the blood and goes to the liver, and the glucagon targets the liver to break down the stored glycogen into glucose, and then glucose is released into the blood. So the blood glucose concentration rises back to normal. This is the normal pathway. We have to state the sequence of events starting from the pancreas, right? So you need to start from the pancreas, and you need to state the sequence of events that are happening after the person does 1 hour of exercise. So because the person did exercise, the blood glucose concentration has fallen. So obviously more glucagon will be released. So this will be 5 and not 6. I told you all, and I usually tell my students, that glucagon is released when glucose is gone. So always remember this point, that glucagon, right? Glucagon is released when glucose is gone. What happens when glucagon is released? Glucagon targets the liver and stimulates the liver to break down glycogen. So there will be less glycogen in the liver because glycogen is broken down into glucose. So the step will be 4. And then because the glycogen is broken down into glucose, the glucose is released into the blood. So now there will be more glucose in the blood. So the sequence of events will be 5, 4, and 1. The answer is B, 5, 4, 1. Let's move on to MCQ number 10. The diagram shows the first part of a kidney nephron and its blood supply. During filtration, protein molecules do not pass through the wall of the glomerulus. Which part contains the highest concentration of protein? So guys, you all know that this is the diagram of the kidney nephron and the associated blood vessels. The part I'm shading right now is the kidney nephron. This is, as you all know, is the Bowman's capsule, right? This is the Bowman's capsule of the kidney nephron, and this is the proximal tubule, right? Proximal convoluted tubule. And these are the blood vessels. This is the afferent arteriole, and these are the capillaries, the glomerulus, and this is the efferent arteriole. So in the question, it is mentioned that when the filtration occurs, the proteins do not pass through. You all know that whenever filtration occurs, small molecules like water, like glucose, like amino acids, minerals, these all pass through, but proteins do not pass through, right? So the blood is flowing like this. However, proteins are not leaking out. The blood is flowing like this, but the proteins are not leaking out. The other molecules are leaking out, especially water is leaking out. So what will happen is that the water is leaking out. However, the proteins are not leaking out. So the concentration of protein will increase as the blood flows through the glomerulus. So the concentration of protein will be highest at D. And why is that so? Because the blood has passed through the glomerulus, which is the network of capillaries, right? As you all know that this is the glomerulus, which is the network of capillaries, and all the molecules are being filtered, especially water is being filtered. So the blood becomes concentrated and the proteins inside the blood become concentrated. So when the blood reaches the point D, the proteins have the highest concentration because a lot of water is filtered from the blood into the nephron. So the highest protein concentration is at D. I hope you understand this. Let's move on to MCQ number 11. Which row describes sexual reproduction? So guys, as you all know, that sexual reproduction is a type of reproduction which involves the fusion of gametes. And gametes are haploid cells, or we can say the nucleus of the gamete is haploid. When the gametes fuse, zygote is formed, and zygote nucleus is obviously diploid because the zygote is formed by the fusion of two haploid gametes. So the zygote nucleus is diploid, and the offspring that are produced are genetically different. These are the features of sexual reproduction. The answer is C. Let's move on to MCQ number 12. The table shows some features of three types of microorganisms. So guys, there are three types of microorganisms, 1, 2, and 3, and their features are shown, and we have to identify that which type of microorganism is which. So if we look at the type of microorganism 1, it does not contain a cell wall, it does not contain a cell membrane, it does not contain a nucleus, it does not contain a cytoplasm. So guys, because it does not contain a cell membrane and it does not contain a cytoplasm, this means that it is a virus, because absence of cell membrane and cytoplasm indicates that there is no cell structure, and this could be a virus. If we talk about microorganism 2. So microorganism 2 has a cell wall, it has a cell membrane, it has a cytoplasm. So it has cell membrane and cytoplasm, it means it has a cell structure, but it does not have a nucleus. So guys, if an organism has a cell structure but no nucleus and also has a cell wall, so we are definitely talking about bacteria. So microorganism 2 is bacterium. If we talk about microorganism 3, it has a cell wall, it has cell membrane, it has nucleus, it has cytoplasm. So this could be a fungus, because as you all know that fungi have cell structure, proper cell structure. They have cytoplasm, they have cell membrane, they also have nucleus, and they have chitin cell wall, right? So answer is C. Let's move on to MCQ number 13. The graph shows the transpiration rates of two plants during one day. Both plants were grown under identical conditions. So guys, there is plant X and plant Y, and their transpiration rates are shown. Which statement explains what the graph shows? So guys, you can see that plant X has the maximum rate of transpiration at which time? Near 12 p.m. That is midday, right? And plant Y has a maximum rate of transpiration near 0000, that is 12 a.m., that is midnight, right? So which statement explains what the graph shows? Guys, you need to know that transpiration occurs via stomata, and maximum transpiration or maximum rate of transpiration means that most of the stomata are open, or we can say all the stomata are open. So plant X closes its stomata during the brightest part of the day. Guys, you need to know that 12 p.m. or midday is the brightest part of the day, and plant X does not close its stomata during the brightest part of the day. Do you understand this? It says that plant X closes its stomata during the brightest part of the day, but we can see that in case of plant X and the brightest part of the day, the transpiration rate is maximum, which shows that stomata are open and stomata are not closed. So plant X is not closing its stomata during the brightest part of the day. Plant Y closes its stomata during the brightest part of the day. This is true. Why is that so? Because we can see that plant Y, if we talk about plant Y has the least transpiration rate near 12 p.m. That is during the brightest part of the day. So plant Y is closing stomata during the brightest part of the day. That's why transpiration rate is the lowest. You should know that transpiration occurs via stomata, and if the transpiration rate is highest, most stomata are open, and if the transpiration rate is lowest, least stomata are open, right? So the answer is B. Let's move on to MCQ number 14. Some people have a rare condition or rare heart condition in which the lower left chamber of the heart, that is the left ventricle, right, has not developed properly and is much smaller than normal. So guys, they are saying that some people have a rare heart condition in which the lower left chamber is the left ventricle, right? Because the lower chambers are the ventricles, and the left lower chamber is the left ventricle. So they are saying that in this rare heart condition, the left ventricle does not develop properly and is much smaller than the normal, right? It's much smaller than normal. The immediate result of this condition is to cause lower than normal blood flow into which vessel? The answer is A, or aorta. That's because you all know that left ventricle pumps blood into the aorta. So what will happen is that this condition will cause lower than normal blood flow into the aorta, because left ventricle is smaller, so it can pump less blood into the aorta? The answer is A. Let's move on to MCQ number 15. Tissue transplants are normally rejected by the body unless the transplant tissue and the patient are very carefully matched. Which component of the blood is responsible for the rejection of transplanted tissue? Guys, the answer is white blood cells. You should know that the transplanted tissues are rejected by the lymphocytes. This is very, very important. Whenever the tissue is transplanted, what happens is that if the tissue does not match, if the transplanted tissue does not match with the antigens of the receiver or the recipient, right, what will happen is that antibodies will form against the transplant tissue, and antibodies are formed by lymphocytes. So lymphocytes are the main cells which are involved in tissue rejection. Let's move on to MCQ number 16. The diagram shows movement of substances between the blood in a capillary and tissue fluid. By which process does the water move from the tissue fluid to the capillary? Guys, we can see over here that the water is flowing from the capillary into the tissue fluid. At which end? At the end where the capillary starts. So capillary starts at arterial end, because over here there is an artery and we call it arterial end. This is the arterial end because over here the artery is ending and the capillary is starting, right? So at the arterial end, the water leaves the capillary and comes into the tissue fluid due to high blood pressure. But if we talk about the other end of the blood capillary, at the other end of the blood capillary, which is known as the venous end, what happens at the venous end? At the venous end, there is low blood pressure. And in the blood over here, there is lower water potential as well. So the fluid returns, the tissue fluid or the water from the tissue fluid returns back to the capillary at the venous end. And this is not because of the high pressure, this is because of the osmosis, right? So you need to know that if water is flowing into the capillary from the tissue fluid, that occurs at the venous end, and this is caused by osmosis. So the answer is C. If they said that what causes the water to flow out of the capillary into the tissue fluid at the arterial end, so the answer would be high blood pressure. The answer is C. Let's move on to MCQ number 17. An experiment is set up as shown. Test tubes 1 and 2 contain lime water. Lime water is a clear solution that turns cloudy. In the presence of carbon dioxide, air is pumped through the apparatus. What is the appearance of the lime water in test tubes 1 and 2 after a period of 10 minutes? So guys, over here, if we observe, we can see that in test tube 1 and test tube 2, there is lime water. And you know that carbon dioxide turns the lime water milky, or we can say is that the carbon dioxide turns the lime water cloudy. Over here, if we see, air is flowing into the first test tube, and the first test tube contains a solution to absorb carbon dioxide. So air contains carbon dioxide, but the carbon dioxide is absorbed from the air by the solution which is absorbing the carbon dioxide. So the air that is leaving this test tube does not have carbon dioxide or does not contain carbon dioxide. So when this air is bubbled into the lime water in test tube 1, the lime water stays clear, right? Lime water stays clear because the air that is flowing does not contain any carbon dioxide, because the carbon dioxide was absorbed by the solution that is absorbing the carbon dioxide, right? Then the air travels from this test tube, that is test tube 1, into the conical flask, and this conical flask contains living animals. These living animals are respiring and releasing carbon dioxide. So the air gets carbon dioxide, air receives carbon dioxide, right? So the air when flows into the delivery tube and goes to the test tube 2, this time the air contains carbon dioxide, and as the air bubbles into the lime water turns cloudy or milky. So the test tube 2, lime water in the test tube 2 will turn cloudy after 10 minutes. However, the lime water in test tube 1 will stay clear. Let's move on to MCQ number 18. Which equation represents anaerobic respiration in yeast? Guys, you need to know that in yeast, the anaerobic respiration is of which type? It's alcoholic fermentation. You need to remember this. So in yeast, The type of anaerobic respiration in yeast, the type of anaerobic respiration that occurs is alcoholic fermentation, in which the glucose is broken down into ethanol and carbon dioxide. The answer is D. If we talk about bacteria, so in bacteria, or we can say in mammalian muscles, the anaerobic respiration is of which type? It's of which type? Yes, guys, lactic fermentation. And in lactic fermentation, what happens is that glucose is simply broken down into lactic acid, right? Glucose will be simply broken down into lactic acid. All right, let's move on to MCQ number 19. The graph shows the rate and depth of breathing at rest. So guys, this is a graph which shows lung volume. Lung volume is plotted on y-axis and time is plotted on x-axis. So how to read this graph? As the lung volume rises, it means that the person is breathing in. So this is inhalation or breathing in, and then this is exhalation. This is inhalation, this is exhalation, this is inhalation, and this is exhalation. So guys, this graph can give you two important informations. And what are those? The number of waves is equal to the breathing rate, right? If the graph spans 60 seconds, so we can calculate the breathing rate, right, by counting the number of waves. Simply, if we count the number of waves in a particular period of time, right, we can use that to find the breathing rate. For example, if there are 3 waves in a period of maybe 15 seconds, for example. So if there are 3 breaths, one wave means one breath. So if there are 3 breaths in 15 seconds, so how many breaths will be there in 60 seconds? That will be 12 breaths. So the breathing rate will be 12 breaths per minute. This is how we find the breathing rate from this graph. And one more thing that we can find is the depth of breathing. So guys, depth of breathing is the volume of air that you breathe in one breath. So this is the depth of breathing actually. For example, initially the lung volume, or initially, for example, the air in the lung was maybe 1500 centimeter cube. And when you took the breath in, the volume is now 2000 centimeter cube. So what is the depth of breathing? It's 500 centimeter cube. So this is the volume of air that you are inhaling per breath, right? So from this graph, you can find the depth of breathing. That is the volume of air that you breathe in one breath, right? The air that you are breathing in one breath is the depth of breathing. And over here, we can see that if the initial volume in the lung was 1500 centimeter cube and the final volume is 2000 centimeter cube, it means the person has inhaled 500 centimeter cube. That is the depth of breathing. And if there are 3 breaths in 15 seconds, then the number of breaths in 1 minute is 12. That is the breathing rate. So breathing rate is the number of breaths per minute, and the depth of breathing is the volume of air that is inhaled in one breath. Now they're asking that which graph shows the rate and depth of breathing during exercise. So guys, you should know that during exercise, both breathing rate and depth of breathing increase. So you need to know that when a person exercises, the person takes more breaths in 1 minute, the breathing rate increases, and the person inhales more air in one breath, or we can say person breathes deeply, so depth of breathing increases. How do you know? How can you figure out that rate of breathing has increased or the depth of breathing has increased? Let me give you an idea. Guys, if the amplitude increases, always remember this point, if the amplitude increases, this means if the amplitude increases, this means that the depth of breathing has increased. And if the frequency or the number of waves in the same period of time increases, it means rate has increased. So how can you figure out that the rate of breathing has increased? There will be more number of waves in the same period of time, right? Over here, we can see that in 15 seconds, there are 3 waves. So find a graph where there are more waves. So in graph C and D, you find more waves. However, the answer is C because in C, you also find a higher amplitude. So always remember this point whenever a person exercises. So what is the change in the graph? There is high amplitude and there is high frequency, right? High amplitude means increased depth of breathing and high frequency means more number of waves in the same period of time and more breathing rate. The answer is C. Let's move on to MCQ number 20. How do the concentrations of oxygen and urea in the renal artery compare to their concentrations in the renal vein? So guys, you should know that renal artery brings oxygenated blood to the kidney. So the oxygen concentration in the renal artery will be higher than that of the renal vein. And you need to know that urea in the renal artery will be higher as well as compared to the renal vein because kidneys are filtering urea from the blood. So the blood is brought to the kidney by the renal artery, right? And the kidney is filtering urea from the blood in the renal artery. So the blood that goes into the renal vein has low urea content. So the urea is high in renal artery and oxygen is high in the renal artery as well. So the answer is A.